Researchers studying at the University of Bologna have estimated there are 37 trillion cells in the human body. These cells communicate with each other through the use of over 50,000 different proteins. Though the study of cells and proteins is basic biology that can be done with a microscope, the magnitude of these interactions demands the need for models and large-scale analyses. Systems biology utilizes state-of-the-art engineering tools to solve complex biological problems. Looking through this lens, there are many cells wet-mounted on a glass slide. The cells can interact with each other through chemical and physical interactions. These chemical interactions include biochemical molecules such as gases, ions, lipids, and proteins. Comparing relative sizes, a virus is to a cell as a cell is to a human. The process in which a virus enters a cell is orchestrated by a complex network of viral proteins working in unison to trick the cell. The infected cell triggers a pathway response dependent on the nature of the virus. Systems biology's approach to tackling this problem is to employ ordinary differential equations as a mathematical model to simulate how these scenarios evolve over time. Solving these ordinary differential equations is not trivial. Just as microscopes are needed to see cells, computers are helpful to understanding them. To appreciate how far computational power has come since the turn of the millennium, consider the expenses it takes to map the entire human genome. According to the National Human Genome Research Institute, the cost per genome in 2001 was $95 million. Currently, the same genome can be mapped at just over $1,000. Systems biologists have expanded their toolset from analytical modeling to include the application of computer science methods. These tools include machine learning, statistical modeling, and graph theory, among others. For example, looking at the activity of 50,000 proteins in the body would be an experimentalist's nightmare. Protein-protein interaction networks allow for the discovery of meaningful patterns in large-scale data and in a fraction of the time. A suitable analogy to this complexity is the mapping of train schedules across the United States. Imagine planning a trip from San Francisco to Boston. No direct route exists, so the train would transfer from San Francisco to Chicago to New York City and then finally to Boston. Now imagine there's a giant blizzard in Chicago, shutting down the train station. The trip would be canceled because there is no route to Boston that does not include Chicago. Similar principles can be applied within the network of protein-protein interactions. Where the stations were connected by train tracks, lines between proteins of a protein-protein interaction graph signify a known interaction from experimental data. As a result, this method infers that proteins that do not directly interact could still have an effect on each other. Similarly to when the Chicago train station was removed from the map, if a virus enters the cell and blocks the creation of a protein, communication within the network can be severely inhibited. Through these various techniques and methods, systems biologists can understand detailed dynamics as well as broad-spectrum interactions occurring within the human body. This information reveals the cause and effect of diseases and allows for development of novel pharmaceuticals. All of these interactions occur at the microscale and smaller. However, the effects seen from these interactions are felt at the macroscale. Therefore, the challenge for systems biologists is bridging the gap between micro and macro scale processes.